So in this last part of our notes, we're going to look at ecological concepts related to big idea number 4. So we see that there's interactions that take place in ecosystems. Ecosystems are very complex, and even minor changes can have dramatic effects on that ecosystem. So hopefully this diagram makes sense. When we first started out the year in biochemistry, we talked about atoms make up molecules, and we just recently reviewed that there's 4 types of organic molecules: the proteins, carbs, lipids, and nucleic acids. And we learned the atoms that are found in each. So proteins have carbon, hydrogen, oxygen, and nitrogen. Carbs have carbon, hydrogen, and oxygen. You mostly just find carbon, hydrogen, and oxygen in most lipids. And carbon, hydrogen, oxygen, nitrogen, and phosphorus in nucleic acids. So it takes these 4 types of organic molecules to make up cells. Similar cells work together to form tissues. There's 4 different types of tissues that we find in organs, and then it takes multiple organs to make up an organ system. So these terms that I have listed for you are just common terms that you don't have to necessarily define, but you need to know what we're talking about when we're talking about a population or a community. So when we talk about a population, that's just a group of similar organisms. So think of like a herd of elephants. When we talk about a community, think about the other organisms that would interact with elephants. So zebras and lions and hyenas, and don't forget the plants that would be in that environment as well. When we talk about an ecosystem, we're basically talking about the community, but we're also factoring in the non-living components. So we're talking about temperature and rainfall in an area. Now, students often get habitat and niche confused. Habitat is the actual area where an organism lives, eats, and reproduces. That niche refers to the job or the role of an organism in an ecosystem. So if you're trying to figure out what an organism's niche is, then think about what could negatively happen if that organism is no longer found in that ecosystem. A niche is basically a job, and so think about what that organism provides or what it does to help maintain stability in that ecosystem. So when we think about a plant or a producer like a tree, then think about what it's providing to that ecosystem. So in this situation, talking about this acacia tree, it's providing food and shelter, and it's also important in maintaining soil moisture. Whenever you think of like birds or frogs, think about what they're supposed to eat, because that's their job, is to control certain populations so those populations don't get out of control. So again, habitat is where an organism lives, and a niche just refers to an organism's job in that ecosystem. Now we're going to get into the 3 different types of relationships. There's competitive, cooperative, which are sometimes called symbiotic, so you can use those terms interchangeably, and then that predator-prey relationship. So in a competitive relationship, organisms are trying to occupy the same niche, or they're trying to utilize the same resources. Now, the competitive exclusion theory states that no 2 species can occupy the same niche for an extended period of time. One species will eventually drive the other one to an extinction. In most ecosystems that are stable, we really don't see this competitive exclusion theory play out, because each organism in that ecosystem has its own niche. But whenever we have a situation where a species is introduced or it's exotic and we accidentally introduce it into that ecosystem, then we see this competitive exclusion theory play out. So a good example is with the native mussels that we have in our freshwater ecosystems in North America, and this introduced species, the zebra mussels. In this situation, the zebra mussels have a considerable advantage. One thing is they're smaller, so they can actually fit in other places that these native mussels can't, which in general are larger. I've even seen some zebra mussels attach onto smaller organisms like crustaceans that live in these freshwater ecosystems, whereas those larger mussels aren't able to do that. And I've even seen situations where these zebra mussels get inside of these larger native mussels. They end up killing them, and then they use their shells for protection. These zebra mussels have a really high reproductive rate, so their population increases exponentially. Another advantage that we see, and this is with most introduced species, there isn't any native predators that are able to eat them. So in this situation, and where we see two different species competing for the same niche, one species is going to have better adaptations. That's going to allow it to be more successful and outcompete that other species. To reduce competition, there's something that's called resource partitioning, or sometimes it's called niche partitioning. So this actually reduces competition. So in this picture down here, you can see that it looks like there's many different birds that are living in the same habitat, but they actually have different niches because it's like they've divided the resources in that habitat. So some birds will feed in the deeper waters, some are feeding in the more shallow waters, and you can see that they all have different adaptations that help them get their resources in those different areas. While we're talking about niche, let's talk about character displacement. We see character displacement, or a change in features, depending on whenever two populations are together versus when they're separate. A good example is with these birds. Whenever you have these birds and they're living in the same environment, you can see that they develop different adaptations. So this Fortis species will have a more deep beak, and this other species has more of a shallow beak. Whenever they're separate, whenever they're not living in the same area, you can actually see that that adaptation seems to disappear. Their beaks aren't as deep whenever they're separate. And for the Fortis species, you can see that their beaks aren't as narrow. So this is what we mean by character displacement. Just simply whenever these populations are together, they're going to have different features than whenever they're separate. Going into our second type of relationship, because remember we're going through different types of relationships in ecosystems. We have predator-prey, we have competitive, and we have symbiotic relationships. So let's talk about the predator-prey. So in a predator-prey relationship, we see the exchange of energy. And in order for it to be predator-prey, it usually ends up in the death of the prey. Those predators are controlling that prey population, and that's a good thing. If it wasn't for predators, then populations of prey would continue to increase, and eventually they become overpopulated. And this is going to lead to starvation and a rapid spread of disease when the population becomes more dense. Now, there was a question on an AP exam a few years ago, and this is an example of one where there's no formula that goes with it. So in this situation, you need to use common sense and know that it's probably going to be a very simple, just mathematical calculation that you have to do. It says, use the graph above to calculate the lag time in months between the change in the densities of the prey and the predator populations. Give your answer to the nearest month. The answer to this question simply was, look at a change, a point in time for that rabbit population, and then find that same type of change in that predator population. You have a maximum amount of rabbits there, and then you have a maximum amount of fox there. We're just looking at that change in time between those two maximums. So in this situation, it's about 2 months. So this was just a simple calculation. It wasn't related to the formula sheet, and it had a tendency to throw students off. Now, predators have adaptations to help them catch their prey, and prey have adaptations to help them keep from being caught. In the evolution unit, we talked about whenever we see the prey population adapt, then you're going to see the predator population adapt as well, and that was coevolution. I'm sure you know what it means if an organism is camouflaged, but this can also be called cryptic coloration. Cryptic means obscure. Mimicry is an adaptation where one species copies another species, and there's two types. First one is called Batesian mimicry, and the next one is called Müllerian mimicry. Now, if I were you, and I would use these little tricks to remember the difference between the two. When I think of Batesian, I think of B for butterfly. So I'm going to give you a classic example with the monarch and the viceroy butterflies. And then think of that M for Müllerian. Think of many. In Batesian mimicry, what happens is we have one species, and it's actually going to copy the look of the toxic species. This is an example of the viceroys and those monarch butterflies. If a bird eats a monarch butterfly, it becomes sick because those monarchs are toxic. Over time, the viceroys that more closely resembled those monarchs were the ones that were surviving and reproducing. And over time, they look very, very similar because the birds weren't sure if they were going to be catching a viceroy or catching a monarch whenever they see this black butterfly flying around with orange markings on it. In this situation, in Batesian mimicry, there's one that's toxic and the other one's not. The one that is not toxic is copying the look of the toxic one. In Müllerian mimicry, there's many species and they're all toxic, and they have that same coloration. So, for example, if you see something yellow and black flying at you, then you have a tendency to think that it's a bee, it's a wasp, it's something that is going to sting you. Since many species have this coloration, its frequency makes it easily learned by predators because so many different species have that coloration. Now, when we talk about deceptive markings, we're talking about things that are confusing to predators, like eye spots. We already talked about agonistic behaviors earlier in that second lecture. These are just behaviors that are threatening to the predators. And then aposematic coloration. If you oppose something, you're against it. So in aposematic coloration, we're against eating those organisms because they're a bright color. And that means that they're toxic in most cases. So it's a warning to predators. That organism shouldn't be eaten. So we have prey adaptations, but we also have predator adaptations, which help them acquire food. So things like venom and toxin as well, and camouflage. And you can usually tell what organisms eat if they're herbivores, if they're carnivores. You can usually tell by looking at their mouth parts, especially with birds. You see a different beak on a bird if they're an insect eater versus if they're a seed eater versus if they're a flesh eater. All right, so on to that third relationship, that symbiotic relationship. Now, you can tell a relationship is symbiotic if it is a permanent relationship between two species. Where you find one, you will always find the other. They do not live apart from one another. And like I mentioned before, sometimes this is called a cooperative relationship, especially when we talk about mutualism. You'll understand why sometimes these relationships are said to be cooperative. So there's three types of symbiotic relationships. There's mutualism, parasitism, and commensalism. Now, if we have a mutual agreement, then we both agree. So mutualism was given to that relationship when both species benefits from the relationship. If I were you, I would probably have at least one example, a real life example of mutualism, parasitism, and commensalism, and have it memorized. Here's some examples for you. So that acacia tree actually provides food for those ants. In return, those ants provide protection. If an herbivore comes and tries to feed on the plant, then they'll attack the herbivore. And they will even prune away other plants that are growing too close to the acacia tree and crowding out the sunlight. We've already talked about plants and that bacteria called rhizobia. So those plants are providing shelter in those nodules, and that the rhizobia bacteria, they're fixing nitrogen into a form that the plants can use. Another type of relationship between plants is called a mycorrhizae. In this situation, we have a symbiotic relationship between plants and certain types of fungi. So let me show you what happens. I'm going to draw a plant cell, but I'm going to show you this fungi, hyphae, that has kind of grown into that plant cell there. It looks like this could be actually more of like a parasitic relationship because we have this fungus, is the fungus that is growing into that plant cell. But actually what's happening is that the growth of this fungus actually increases the surface area where that plant cell can exchange nutrients. So now the surface area extends all throughout that cell. So what's happening in this situation is the plant is producing carbohydrates that the fungus can utilize, and then that plant has greater surface area for the exchange of nutrients and water. So it's a win-win situation for both species, so it's considered mutualism. When we have a parasitic relationship, we have one that's going to benefit, and one is being harmed. So this isn't really necessarily considered a cooperative relationship. So a good example is between a mammal and a tapeworm. Now, the organism that is being harmed or being fed off is called the host. That parasite is getting energy from that host, but it's not considered predator-prey because usually the host does not die. As a matter of fact, parasites don't want to kill the host because they're living off the host, and if the host dies, then the parasite dies. So remember pretty much your bloodsuckers. Those are going to be parasites, so your ticks, your mosquitoes, lice. There are some plants, like mistletoe is actually considered a parasite because it grows into other plants that it's growing on, and it actually takes some of the carbohydrates out of the phloem that that plant makes through photosynthesis. But there's even small organisms, animals like the candiru fish, that are considered parasites. They actually jab themselves into the gills of bigger fish, and they suck the blood. In commensalism, we have one species benefiting, and notice in all three of these symbiotic relationships, one organism benefits. If one didn't benefit, then that relationship wouldn't have developed in the first place. We always have one benefiting, and in this situation, the other is not helped nor harmed. So as this organism goes about its daily business, there's another species that's going to get the benefit of it just going through its normal activities. So a good example is with these different birds, like the egrets, that ride on the back of grazing mammals. Buffalo and zebras and lots of different organisms have these birds associated with them. What happens as those herbivores are moving around, they stir up insects, and these birds fly down and they grab the insects. So this is an example of commensalism. It's not hurting the grazing mammals, it's not helping them. If the birds were feeding off of the parasites that were on those mammals, then it would be mutualism, but in this situation, it's just commensalism. We're going to look at the continued flow of energy from one organism to the next. And we already looked at the movement of energy in that first lecture because we talked about photoautotrophs and chemoautotrophs are able to collect free energy and convert it into organic molecules, where they use that energy to fix carbon from the atmosphere into organic molecules. Often confused, but a simple concept are the arrows in a food chain. Those arrows represent the movement of energy. When you see arrows, this simply means that the plant's energy is going to go to the insect when the insect consumes the plant tissue. The insect's energy is going to go into the shrew. The shrew's energy is going to go into the hawk. So you might envision that arrow going into the mouth of the organism that is eating it. So the plant's going into the insect's mouth, the insect's going into the shrew's mouth, and so on. These are easy concepts, but often missed by students just because they don't take their time to see how these organisms are classified. Whenever we're talking about food chains or food webs, sometimes we'll refer to the secondary consumer or the primary carnivore, and you need to understand what organism we're talking about. We can classify these organisms as producers and consumers, and then we can even classify them further as first level, second level, third level. And of course in science we give everything 2 to 3 names to confuse you all. So if I'm referring to that first level consumer, I can also call it the primary consumer. If I'm talking about the second level, I can call it the secondary consumer. If I'm talking about the third level, I can call it the tertiary consumer. So if I ask you in that food chain, which organism is the primary consumer, you want to start at the bottom of that food chain with that plant, and you want to move up that food chain. So that plant is considered a producer, so it doesn't count. So this insect is the first consumer. The shrew is the second consumer. And the hawk is the third or the tertiary consumer. So that primary one would be the insect. Secondary shrew, and tertiary would be hawk. You need to note whether we're asking for a consumer or if we're asking or referring to the different levels of carnivores. So again, I can say first level carnivore, I can say primary carnivore, secondary, and again tertiary carnivore, which means third. Again, take your time. So when we look at this food chain, we note that the plant is a producer. That insect that is eating this plant is considered an herbivore. So that first carnivore, that primary carnivore, is the shrew. Often students are going to say the primary carnivore is the hawk because they think the biggest organism, that must be the primary consumer, but that's not the case. You've got to start with the producer and you got to work your way up your food chain. That primary carnivore in this situation would be the shrew. The secondary one would be considered the hawk. And in this food chain, we're not showing a tertiary carnivore. Looking at this food web, let's see if you can identify an organism that would be considered both a secondary and a tertiary consumer. So if you want to pause and try this on your own, go for it. Again, we're going to start with plants, and we're going to start moving up the different food chains in this food web until we find one that is both a secondary and a tertiary consumer. So in this situation, we have a producer, and we have, this would be our first level consumer, and this would be our second level. If we go this way, this would be a third level or tertiary consumer. But I can also follow this food chain. And in this situation, again, I have those insects that are eating plants are those first level consumers. In this situation, the spider is the second level consumer. So that would be your answer. The spider is considered both a secondary and a tertiary consumer. A food web just shows many different food chains all linked together. It shows the different relationships between organisms, either directly or indirectly, and it shows that all organisms are interdependent in one another. If you see a change in one, then that's going to affect other organisms in that ecosystem. Be prepared to talk about how this honey badger would be affected if an insecticide was sprayed and it killed off a large proportion of the grasshoppers. Well, you can see that the honey badger isn't directly linked to grasshoppers, but if this was to occur, then there would be a drop in this cardinal population. And so our mountain lion here, won't have this as a food source. And is actually going to probably switch over hunting more snakes and hunting more honey badgers to acquire enough energy. So even though that honey badger isn't directly connected with the grasshopper, it's going to feel the effects if something does happen to the grasshopper population. These food webs also show the complexity of ecosystems or the degree of biodiversity. Biodiversity just refers to the number of organisms in an area. And if you count up the organisms in that area, then you would see that there's 11. Now, in ecosystems, we actually want more biodiversity. The greater the biodiversity or the more complex that community, the more stable it is. And that's usually due to the case that there's more organisms, then there's more food sources for organisms. So if something happens to one food source, then they can prey more on their other food source. So let's take a second to look at a question that talks about biodiversity and the stability of communities. This was a question from the 2013 test. So it says, a researcher is investigating the relationship between the existing species diversity in a community and the ability of an introduced non-native species, so we know this is a negative thing, that can destabilize the community. Which of the following graphs is most consistent with the claim that communities with high diversity are more resistant to change? And that's a good thing. High diversity, we want them to be more resistant to change. We can tell from the question there's going to be a change. We can automatically throw out that first graph because that line isn't showing a change. So let's go down here and let's look at these two graphs that are opposites of one another, C and D. And let's make some notes so we really understand what this graph is showing us. We would have high species diversity on this side, and we have low species diversity. And so it looks like when we have high diversity, which is good, we want high diversity, you can see that we're getting a whole bunch of the invaders surviving, and that's not going to be a good situation for this community. If we look at D, we see that there's high diversity, there's a really low number of invaders surviving. And that would be good for the ecosystem. That means the ecosystem is very stable if an introduced species is not destabilizing it. This diagram that you're seeing right here, this is referred to as an ecological pyramid, but we can also call it a biomass pyramid, an energy pyramid, and a number pyramid. Each level can be called a trophic level or a feeding level. It means the exact same thing. So this is the first trophic level. So we're going to start at the bottom and we're going to go to the top. So this is the first trophic level, or the first feeding level. This is going to contain our producers, our plants, our autotrophs. And that second trophic level, or feeding level, we have the herbivores. On the third, you're going to have your first level carnivores, or your primary carnivores. There's usually even a fourth or a fifth trophic level. A fourth would have our second level carnivores. So if I asked you what trophic level would contain the second level consumers, then you should be able to tell me that the second level consumers would be on that third trophic level, because we have producers. The herbivores would be the first consumers. Those first level carnivores would be our secondary consumers. So the shape of this pyramid is significant. It's wider at the bottom and it's more narrow as we get at the top. Because of this shape, we can conclude that that first feeding level has the highest number of organisms. More than any organism, you're going to have more producers than herbivores, more herbivores than carnivores, more first level carnivores compared to your second level carnivores. So the number of organisms is going to decrease as you move up that ecological pyramid. That first feeding level also has the highest amount of biomass. Now, we're determining biomass of fruit by removing the water. So that's how you calculate biomass, is you have to remove all the water from an organism. And what is left is just going to be organic molecules. And that's what we mean by biomass. So if you dried all the producers in an area versus all the herbivores, you're going to see way more biomass has accumulated in those producers than in those herbivores. And we can also conclude that the first feeding level has the highest amount of energy stored in their tissues. So if we could calculate the amount of calories that would be tied up in plants, it's going to be significantly higher than the energy, the calories that are stored in the bodies of the herbivores, and so on as you move up that energy pyramid. There's a very low amount of energy that actually makes it from one trophic level to the next. And this is referred to as that 10% energy transfer rule. So if there's 10,000 joules of energy tied up and stored in the plants, only 10% of that actually makes it and becomes stored as the tissues in those herbivores. Only 10% of the energy of the herbivores actually makes it up to that third trophic level, and so on. This is why your upper level carnivores, they don't die of predators eating them. They die of starvation because there's just not a lot of energy available to them. This low energy transfer rule just explains why the amount of energy, biomass, and the number of organisms in each trophic level decreases as you move up that pyramid. So let's make sure you can read this diagram and draw some accurate conclusions. How much energy is available to the tertiary consumers? Well, first you have to figure out what we're talking about when we're talking about tertiary consumers. So this is our first consumer, here's our second consumer, and here's our third consumer. So they're talking about how much energy is available to the snake. This is its food source. It only has 100 joules of energy available to it that it has the opportunity to possibly catch. When an organism consumes food or acquires free energy, like we said, only 10% of this energy is actually stored on that organism. And the other 90% is being lost somehow. So we're going to talk about how energy gets lost from food chains. Some of that energy that is consumed is going to be broken down so that the organism can make ATP. ATP provides energy. Whenever that organism uses the ATP, when they use that energy, the majority of it gets converted to heat, and that heat is lost. So some of that food energy is used and it's lost as heat. Now, some of that energy, or some of those molecules that you acquire in your food, are not digestible, but they do have energy in them. So some energy is lost as waste. Since 90% of the energy is lost as we move from one trophic level to the next. That explains why there's not very many trophic levels. There's just not enough energy making it up to continue to support more and more levels. So there's usually only about 4 or 5 different trophic levels found in an ecosystem. Let's look at this diagram and just put that all in perspective. If this caterpillar is eaten by this bird is going to acquire 100 kilojoules of energy. Now, out of all that energy, that bird is only going to store about 10% of it. It's going to store about 10 kilojoules of energy in proteins and carbs and lipids that make up the tissues of the bird. A major portion of that energy is going to be used. So the bird's going to use some of that energy, and it's going to be released as heat. And some of that caterpillar is not going to be digested, and it's going to go right through that bird as waste. So energy is going to be lost as waste. Since we're talking about organisms acquiring energy, storing that energy, using some energy, it's a good time to talk about primary productivity. Now, when we're talking about primary productivity, we're talking about plants, not animals. Primary productivity just refers to the rate at which plants are able to take CO2 out of the atmosphere and convert it into organic molecules. And again, that's done through the process of photosynthesis. So, and plants are taking the CO2 from the atmosphere and they're converting it into organic molecules like glucose and proteins and lipids. Now, before we fill in these blanks here, I think I'm going to just refer to this diagram down below. We know that plants are absorbing energy from the sun, and so all of the energy that this plant is able to absorb is referred to as their gross primary productivity. So I want you to think of gross primary productivity as meaning the total amount of energy that this plant receives and absorbs. Now, we know that plants do respiration as well. They're going to convert some of those organic molecules into ATP, and they're going to use ATP. So some of this energy that this plant accumulates is going to be lost due to a respiration. So what that plant doesn't use, it's going to store. And so the energy that is stored is referred to as that net primary productivity. So if you manage your finances, then you maybe understand more than others what we mean by gross and net. So if we talk about your gross salary, what we're talking about is the total amount of money that you made that your employees pay you. But you know that you're going to have taxes taken out of that. So the government's going to use some of that money, and what you have left over is referred to as the net. That's what you get to keep. That gets to go into the bank account. Okay? So again, gross refers to that total amount of energy that is collected by the plant. Net is what's not used out of that and what is stored. And then energy lost due to respiration makes up the other part of that energy that was collected. Let's take a look at some equations related to gross primary productivity and net primary productivity. Hopefully this diagram will make these equations make more sense. So again, gross is total, and then out of that energy, some of it's going to be lost and some of it's going to be stored. So the equation to calculate gross primary productivity is just adding what was stored, the net, and what was lost due to respiration. And those two, the net and respiration, should equal the amount of energy that was received by that plant. Now from there, you should be able to manipulate this equation. So if they give you gross and they give you net, then you should be able to determine what was lost due to respiration. So to solve for respiration, if you subtract net from both sides, then if you take gross primary productivity, you subtract the net, then you're able to determine the amount of energy that was lost due to respiration. So if you want to solve for net, then you can subtract respiration from both sides. So gross primary productivity, you subtract respiration, then you can calculate the net. So now that you know a little bit about gross and net primary productivity, let's relate that to this diagram. This diagram was off of a gridding question from a past test. Looking at the first question, it says, which amount represents the gross primary productivity of the forest? In this diagram, we see that we have energy coming from the sun. Looks like some of it's being reflected, some of it's being transmitted. And then it's saying that this is the amount of energy that is actually being absorbed by the plants in this hardwood forest. So the gross primary productivity of this forest would be 1.5 times 10 to the 6th. Which amount represents the loss due to respiration? So we know plants are going to convert some of those organic molecules that they made in photosynthesis to ATP, and that energy is going to be used. So this represents the amount of energy that's lost due to respiration, 1.4 times 10 to the 6th. Says which amount represents the net primary productivity of the forest. So this is the total going in. This is the amount that's lost due to respiration. So this is the amount of energy that's left over that's going to be stored as organic molecules on the plants, or you can say that that energy is accumulated in the biomass. So that's the answer to number 3, 1.9 times 10 to the 4th. Number 4 says calculate the amount of energy stored in the shrub layer. It's kind of like whenever you saw the diagram with the flow of carbon through an ecosystem, and I said what comes in must go out. That's kind of the same principle behind this diagram. If we have this much energy tied up in the plants, then this much energy is divided between the 3 types of plants that we find in this hardwood forest. There's some trees, there's some shrubs, and there's an herb layer there. So how much energy is tied up in the shrub layer? When we add up these 3 amounts, it should equal the whole, 1.9 times 10 to the 4th. Now, if this is difficult for students because it's in scientific notation, then feel free to convert it. So this number really means 19,000. This means 13,000. This means 1,100. And again, how you do that is if you write 1.9, 10 to the 4 just means move your decimal over 4 places. So 1, 2, 3, 4, and fill in your zeros. So that's how I got 19,000 out of that. So when you do a little bit of math, if you subtract 13,000 and 1,100 from 19,000, then you're going to see that there was 4,900 kilojoules per meter squared tied up in that shrub layer. In the next part, we're going to take a look at calculations related to a lab that we're going to partially do. Basically, in this lab, what you're expected to do is we're going to hatch out caterpillars. We are going to determine how much biomass they consume. We're going to be able to calculate how much energy they got from eating that amount of biomass. We're going to be able to calculate how much energy they store, they use and lose as respiration, and how much they wasted. So let's look at some example problems. It says students grew Wisconsin fast plants. On day 7, we pull up the plants, we brush off all the dirt, and we mass them. The mass of 10 plants is 19.6 grams. Then what we do is we put them in a dehydrator, and we're trying to determine the mass that's just due to biomass. So we have to remove all the water. And after we remove the water, then we have 4.2 grams. So this is the biomass of those 10 plants. Question number 5 says calculate the net primary productivity for each Wisconsin fast plant per day that was grown. Well, here's an energy conversion for you. We have 4.2 grams of biomass, and it says that in 1 gram of biomass, there's 4.35 kilocalories. We're going to write 4.35 kilocalories is in 1 gram. Our units are going to cancel, and when we calculate that, that means that we get 18.1 kilocalories is the amount of energy that is found in those 10 Wisconsin fast plants. Now, the question asks for primary productivity, so the amount of kilocalories per day per plant. So in 10 plants, if we had 18.1 kilocalories, They took 7 days to grow, so we're going to divide by 7 days. And that was 10 plants, so we need to divide by 10 plants. And when we do that, we're left with each plant stored 0.25 kilocalories per day. And that's per plant. Now, in this lab, we're going to feed the Wisconsin fast plants to the caterpillars. So when the caterpillars are eating the Wisconsin fast plant, they're actually consuming water as they consume organic molecules. So we actually have to be able to calculate the amount of water that's in tissues, or what percent of that tissue is considered biomass. So 6 says calculate the water percent of the 10 plants. When we look at the change in mass, we started off with 19.6 grams, and after we took out all the water, we were down to 4.2 grams of biomass. This means that we lost 15.4 grams of water. To calculate the percent of plant tissue that is water, then we're going to take that water loss was 15.4 grams. Initially, the mass of those plants was 19.6 grams. Before we dried them, we get 0.79. So that means that 79% of those Wisconsin fast plants was actually water, which means that 21% is due to biomass. So we're going to need that to answer question number 7. It says a caterpillar ate 2.5 grams of a Wisconsin fast plant. Using the percent biomass calculated, determine how many grams of biomass were consumed. We know that that caterpillar ate 2.5 grams of Wisconsin fast plants. But again, it's consuming water and it's consuming organic molecules. And we're just interested in the grams of organic molecules it's consuming, because that's where the energy is at, that caterpillar can utilize. There's no energy stored in water that the caterpillar can utilize. So there's 2.5 grams consumed, and we know that out of that 2.5 grams, only 21% of that is actually biomass. So when we do that calculation, those caterpillars only consumed 0.5 grams of biomass. They didn't get 2.5 grams of biomass. Some of that mass that they consumed was water. We just calculated that the caterpillar ate 0.5 grams of biomass. So now we're actually able to calculate how much energy was consumed. Now, up on that table, it says that in 1 gram of plant biomass, there's 4.35 kilocalories. So there's our conversion factor. So in 1 gram of Wisconsin fast plant biomass, there's 4.35 kilocalories. If that caterpillar ate 0.5 grams of biomass, then it actually consumed 2.2 kilocalories. Now, before we get into some of these other calculations, let's go down here to this bottom diagram. And we basically just calculated that from it eating 2.5 grams of the Wisconsin fast plants, or the reason why there's a Brussels sprout there is you can also put these caterpillars on Brussels sprouts, and they'll eat Brussels sprouts as well. They consumed 2.2 kilocalories. So that's what went into the caterpillar. During this time that the caterpillar was eating, we are expected to collect their waste. We're going to dry it and we're going to mass it. And so we're actually going to determine the amount of biomass that passed right through that caterpillar. And that waste, that biomass in the waste contains calories. So we're basically going to be able to calculate how much energy just passed right through that caterpillar from what was eaten. The waste of a caterpillar is called frass. So when I say frass, I'm talking about the waste of caterpillar. It says that after we dry the frass, we have 0.2 grams. And since we dried it, that's the biomass. So there was 0.2 grams of biomass in the frass. And in biomass, there's 4.76 kilocalories per gram. So there's your conversion factor in 1 gram. So our grams are going to cancel. And so we determined that 0.95 kilocalories was lost as waste. So we'll go put that on the diagram here in just a second. We're going to figure out how much energy was stored on the caterpillar from what was taken in from that 2.2 kilocalories that was taken in. So it says that during this time, the caterpillar's mass increased from 12 grams to 12.25 grams. That caterpillar went up 0.25 grams. Now, some of that is organic molecules, biomass stored, and some of it is due to water. So it says, knowing that 40% of a caterpillar's mass is biomass, which means 60% is water, calculate the amount of biomass that was formed and stored on that caterpillar. 0.25 grams, that includes water and biomass. We know that 40%, I'm going to put a line there, just 40% of that amount is going to be biomass. So when we multiply those together, that means that that caterpillar only stored 0.1 grams of biomass on its body. How many kilocalories was stored on that caterpillar? So if we know 0.1 grams of biomass was stored, and there's 5.5 kilocalories in 1 gram of caterpillar biomass, then that means that 0.55 kilocalories of energy was stored on that caterpillar. So let's add those to the diagram. So we know there's 3 fates of energy that is consumed. Out of this 2.2 kilocalories that was consumed by the caterpillar, some's going to be stored, some is going to be released as waste, and then some is going to be used by the caterpillar. We just determined that there was 0.95 kilocalories released as waste. We know that 0.55 kilocalories are stored on that caterpillar as proteins, carbs, and lipids and nucleic acids. So now with that, we can actually calculate the amount of energy that was lost due to use, lost due to respiration. When we add these two together, we get it's 1.5 kilocalories. We had 2.2 go in. We know that 1.5 kilocalories was lost in waste and was stored on the caterpillar. So we're left with 0.7 kilocalories was what was used by that caterpillar and released into the atmosphere. We actually add up what was used and released as heat, stored, and lost as waste, then that should equal the total amount of calories that was consumed by the caterpillar. Last concept that we're going to look at relates to primary productivity of plants, this classic lab that we used to do, and there's a couple calculations on your formula sheet that you need to understand. Again, when we talk about primary productivity of plants, we're referring to the rate at which they're taking in light energy and they're making those organic molecules. And again, that's done by photosynthesis. Remember, for any reaction, we can actually determine the rate of reaction by measuring the amount of the reactants, and these are the reactants. We can measure the amount of reactants that are used or depleted from the system, or we can also measure the amount of products that are being formed. Okay, so we can get the amount of products being formed over time, and that's actually going to allow us to calculate the rate of photosynthesis. What we did in a past experiment was we filled up bottles with algae and water. And what we did after that is, for some of these bottles, we wrapped them in screens. So what we were doing is we were limiting and we were changing the amount of light that was going to be absorbed by algae. And algae is a plant that does photosynthesis. After we had the bottles wrapped, and we completely wrapped one in foil, and we left one completely unwrapped. So this bottle represents the bottle that's going to receive 100% of the light. We actually measured the amount of oxygen that was present in that bottle initially before we placed these bottles underneath the light. So we placed these bottles underneath the light for about 24 or 48 hours. You're going to need to be able to read experiments, understand what's going on, and be able to predict what's going to happen or be able to draw conclusions from the data. So just from that experiment description, let's see if we can answer these questions. It says, which bottle should contain algae that would gain the most biomass after 24 hours? Well, we know that this bottle is receiving 100% of the light. There's going to be a higher rate of photosynthesis. So that means that the plants are acquiring more energy to convert CO2 into organic molecules, which is what we mean by biomass. So that bottle that's getting 100% of the light should gain the most biomass after 24 hours. Which bottle will contain the most amount of oxygen in it after 24 hours? Photosynthesis is making organic molecules, and it's also making oxygen. So if photosynthesis is occurring, then O2 levels should be going up as well. And so we should again see more oxygen in that bottle that's getting 100% of the light. Which bottle will contain the least amount of oxygen in it after 24 hours? Plants do photosynthesis, but they also do respiration because they have to convert those organic molecules into ATP and power the cell processes. We know respiration actually uses oxygen from the environment. In this bottle, respiration is occurring, and we should see oxygen levels go down as respiration is occurring. The bottle that's receiving 0% of the light, or the bottle that's completely covered, should contain the least amount of oxygen after 24 hours. And then it says which bottle would show you the amount of oxygen consumed in respiration. Well, we can actually look at the change in oxygen. Let's say that this bottle started off with 4 parts per million of oxygen. And at the end of the experiment, when we measured the amount of oxygen, it was down to 2 parts per million. Went down to 2. We can actually calculate how much oxygen is consumed in respiration. And we can use that bottle again that's not receiving any light. On your formula sheet, these are the 2 equations that are related to primary productivity in plants. Basically, what we're able to calculate is how much carbon was fixed or made into organic molecules through the process of photosynthesis. Let's take a look at this bottle that was getting 100% of light. Remember that we measured the initial oxygen concentration. There was 4 parts per million of oxygen in that bottle. This first example says that after we measured the oxygen levels, we determined it was 8 parts per million. So it increased by 4 parts per million because of photosynthesis. I'm using parts per million, and if you look at our equations here, there's no units that are parts per million. They would have to actually give you this conversion because this is not something that is on your formula sheet, or it's not anything that you have to have memorized. So this problem says for 1 part per million of oxygen, there's 1 milligram of oxygen per liter. When we look at these units right here, we can see that it actually matches these units. The question says, how much carbon was fixed by the algae through photosynthesis in that 24-hour period if 4 parts per million of oxygen was made during those 24 hours? We're trying to get here. We're trying to figure out how much carbon was fixed. Well, we're going to just simply use these 2 different equations. We're going to do 2 steps, and eventually we're going to calculate how many milligrams of carbon was fixed in photosynthesis. We have 4 parts per million, but we know that 4 parts per million is going to equal 4 milligrams of oxygen per liter. And that is, again, this part of the equation. So we're just going to plug in 4 milligrams of oxygen per liter. We're going to multiply it what the formula tells us to multiply by, which is 0.698. Let me move my screen up a little bit. 0.698. And that gives us 2.8 milliliters of oxygen per liter. Gives us that number right there. Once we have that number, we can actually plug it into the next equation. So if we have 2.8 milliliters of oxygen per liter, we're going to multiply that by 0.536, like the equation shows us. And that tells us that during that time, 1.5 milligrams of carbon was fixed. So let's just do one more problem to practice working with those equations. It says, if 2 milligrams of oxygen per liter was produced, how many milliliters of oxygen does that equal? So if you look at the units, that's just that first equation. So we're going to just plug in our numbers. 2 milligrams of oxygen per liter. We're going to multiply by 0.698. Gives us 1.4 milliliters of oxygen. And then A says, how many milligrams of carbon per liter were fixed? Which is just that second equation. So we're going to plug in the 1.4 milliliters of oxygen per liter. The equation says to multiply that by 0.536. So we can tell that there was 0.75 milligrams of carbon fixed during those 24 hours.